Hello, everyone. Today we will be talking about ATP synthesis. We will be breaking this topic down into the electron transport chain and ATP synthase. So part one, the electron transport chain. First off, the electron transport chain takes place in the mitochondria. So things to know are the intermembranous space, the intermitochondrial membrane, and the mitochondrial matrix. And before we get into it, there's just some vocabulary that I think we should go over real quick. So first of all is the reductant, which is the reduced form of a species, and the oxidant, which is the oxidized form of a species. Next, a complex is made up of subunits. And complexes in the electron transport chain each have their own set of amino acids that act as electron acceptors and donors. And if we remember the definition of acids and bases, are electron donors and acceptors. Ubiquinone is another term that we'll be hearing, and that is the oxidized form of coenzyme Q. And the reduced form of coenzyme Q is ubiquinol. Cytochrome C is a carrier that we'll be talking about, and it has a heme group. And in this heme group, the iron 3+ carries one electron from complex 3 to complex 4, which means that it's reduced at complex 3 and oxidized at complex 4. So first we'll start off by talking about the NADH electron transport chain. There are 3 complexes in this chain. First is complex 1, and then 3, and then 4. In complex 1, the reductant is NADH, and the oxidant is ubiquinone. In complex 1, 4 hydrogens are pumped across the membrane. In complex 3, the reductant is ubiquinol. The oxidant is cytochrome C, and 4 hydrogens are pumped across the membrane. In complex 4, we have cytochrome C as our reductant, oxygen is our oxidant, and 2 hydrogens are pumped across the membrane. In total, we are pumping 10 hydrogens across the membrane, and this will be important later on. So let's look at exactly how this happens. So here we have a little diagram of the electron transport chain for NADH. So first, what happens is that NADH is converted to NAD plus H. And when this happens, ubiquinone, or Q, is converted into ubiquinol, or QH2. And for this to happen, we have to have 2 protons come and join the ubiquinone to make ubiquinol. And while this is happening, 4 hydrogens are pumped up across the membrane through complex 1. So this is what we have at the end of the first step. We have the 4 hydrogens that we've pumped across the membrane, the NAD plus H plus that we've created, and the ubiquinol that we've created as well. So in the next step, we're going to convert this ubiquinol back to ubiquinone. And the electrons that are released from this process will go up into the cytochrome C. These 2 hydrogens are going to be released across the membrane, as well as 2 more hydrogens from down there. So the extra electrons that we've just added to the cytochrome C are going to prompt it to move over to complex 4. At complex 4, it will release its electrons, and this will aid in the reaction that turns oxygen plus 2 hydrogens into water. At the same time as this is going on, 2 hydrogens are released across the membrane. So that was the NADH electron transport chain. Next, we'll talk about the FADH2 electron transport chain. So in this one, just like in the other one, we have 3 complexes that we're looking at, but this time they are 2, 3, and 4 instead of 1, 3, and 4. In complex 2, the reductant is FADH2, and the oxidant is ubiquinone. In this one, 0 hydrogens are pumped across the membrane. Complexes 3 and 4 are the same as earlier. So in this one, we have only 6 hydrogens pumped across the membrane. So what happens first is that FADH2 is converted to FAD, and ubiquinone is converted to ubiquinol. And then we have the same reaction as last time. Ubiquinol is converted back to ubiquinone. The electrons move up into the cytochrome C, and the hydrogens, as long as 2 hydrogens from down here, are released across the membrane. The cytochrome C transfers over to complex 4, and water is created, as well as 2 additional hydrogens being pumped across the membrane. Part 2, ATP synthase. ATP synthase is the enzyme that synthesizes ATP. It has a lot of parts, so we'll break them down here first. We have the A subunit, and this is the place where hydrogen enters ATP synthase. The A subunit passes the hydrogens to the C subunits. And each ATP synthase has 8 to 15 C subunits. Each C subunit has a hydrogen bonding site, which is where the hydrogens go after they come in from the A subunit. When the hydrogens come into the C subunits, they rotate around, which we'll get into more detail later. And so this rotation allows them to release hydrogens on the other side of the membrane, which is the matrix side. The gamma subunit rotates due to the rotation of the C subunits. And then finally, beta subunits are where ATP is actually synthesized. So first we'll look at the C subunits, which are these little yellow dots, and the orange dot is the gamma subunit. So if we look at this A subunit, we can see that the hydrogens come in on the intermembrane space side, and the exit on the matrix side. And here's how it kind of rotates. Watch the C subunit that has the black outline. So it goes around in the circle, and as it's doing that, the gamma subunit is also rotating. And it's really because of that. So the hydrogens come in here, they go all around, and then they leave here. And this kind of reminds me of those human-powered Ferris wheels. When you get on, it goes all around until it's off. And the people getting on and off are what cause the rotation of the Ferris wheel. So next we'll talk about the beta subunits. There are 3 conformations of beta subunits, which are caused by the rotation of the gamma subunit. These conformations are loose, tight, and open. In the loose conformation, ADP and PI are bound to the beta subunit. In the tight conformation, ATP is favored, which is unique for an enzyme because usually they favor the transition state. But because the creation of ATP is an unfavorable reaction, it needs to be a different kind of enzyme. So the open conformation is the conformation in which ATP exits the beta subunit and a new ADP and phosphorus bind. Here we can see the beta subunits. We can see that in the tight conformation, we have this reaction that turns ADP and PI into ATP and water. We can see in the loose conformation, we have ADP plus PI, and in the open conformation, we have ADP leaving the beta subunit and ADP plus PI entering. And so as the gamma subunit turns, we also see that which beta subunit is T, L, or O also changes in response to the gamma subunit turning. So here we can see that this one up top on the left is the tight conformation. And when the gamma subunit moves, the tight conformation is suddenly this one on the bottom. So 3 ATP are synthesized per 1 complete rotation of the gamma subunit. So basically, 1 ATP is created at each beta subunit per complete rotation of the gamma subunit. And that's ATP synthesis. Thank you for watching.